Hello, scientists. We have with us today Dr. Qingwu Meng, the lead researcher who generated the experimental results that you've been deciphering. How about we hear directly from him about what the data means? Dr. Meng, would you please walk us through the different treatments that you set up for this experiment?
Yeah, of course. Thanks for inviting me, Dr. Williams. In this experiment, I looked at how lettuce and kale grew indoors under different combinations of blue, green, red, and far red light, as well as two white light spectra. In this figure, B is blue, G is green, R is red, FR is far red, WW is warm white, and EQW is equalized white. There were 10 lighting treatments in total. Under the photo of each treatment, there is a treatment symbol as a pie chart. This provides a visual breakdown of light colors. Next to the symbol is the treatment name. The number after each light color shows its photon flux density or brightness in a unit called micromoles per meter square per second. It tells us how many photons, which mean light particles, fall on a square meter in a second. The higher this number is, the brighter the light is. For example, the top left treatment is B60 R120. This means it had 60 micromoles of blue light and 120 micromoles of red light at the plant level. Below the treatment name is a spectral distribution curve. The x-axis is light wavelength. The y-axis is photon flux density. Blue light is photons between 400 and 500 nanometers. Green light is photons between 500 and 600 nanometers. Red light is between 600 and 700 nanometers. And lastly, far red light is 700 to 800 nanometers. The area under each curve is the total photon flux density. In this experiment, our light sources were light-emitting diodes, or LEDs, as you can see in these pictures. The LEDs have distinct peaks for each light color. Okay, now let's talk about the treatment design. I had a blue and red treatment as a control. Additionally, I substituted blue light with green light or far red light at 20, 40, or 60 micromoles while keeping the total photon flux density and red light the same across all treatments. I also delivered a treatment with equal blue, green, and far red light. Lastly, I included two white LED treatments with different spectral distributions that covered a broad range of wavelength.
Wow, this figure really summarizes the plant responses to the light quality treatments. What were the key findings?
So for each crop, I grew multiple plants under each treatment, and I also repeated the experiment over time. I took a photo of an average-looking representative plant from each treatment. And now I will explain a few key findings. In this chart, bars indicate leaf length, whereas the squares indicate shoot dry weight or biomass. The first key finding is that as the proportion of green light or far red light increased, leaf length or plant size increased for green leaf lettuce Rex. This translated into higher yield or biomass because plants with larger leaves captured more light for photosynthesis. We also observed yellower and more spindly leaves under far red treatments than without far red light. On the other hand, red leaf lettuce Ruchii showed similar responses in terms of morphology, which means plant shape, and biomass, which means yield. Interestingly, increasing the proportion of green light reduced red leaf coloration. So under this treatment with only red and green light without any blue light, red leaf lettuce actually looked like green leaf lettuce.
Wow, such nifty results. Plants are amazing. Dr. Meng, why is this research so important to the horticulture industry?
There are more and more people interested in urban agriculture and indoor vertical farming, especially in urban areas. So we can provide fresh, local, and nutritious vegetables to city dwellers. This new type of farming has precise environmental control and saves on space. However, it relies on LED lights to provide the light that plants need to grow. Our research can help us understand how different crops respond to different light qualities. And the results can help growers select optimal light combinations to improve crop yield and quality. So growers can increase plant productivity and profitability.
Awesome. Thank you so much for sharing your very interesting research with us.
Thank you, Dr. Williams.